Morning, students. Today we are going to discuss the next topic, that is mycorrhiza from Unit 4, and it is from the subtopic of scope and applications of microbes in biotechnology and specifically agriculture. So mycorrhiza, what are they? Let us have a small introduction about mycorrhiza. These are basically, mutualistic and symbiotic associations which are formed between the roots of higher plants and the fungi. So when you say mycorrhiza, it is actually a Greek word which is formed from two words, that is myx, which means mushroom or fungi, and rhiza means roots. So mycorrhiza is actually a association between the roots of a higher plant and the fungi. So it is a symbiotic association wherein both the members in the association are having a mutually favorable relationship with each other, and no one is harmed because of this relationship. So it is a mutualistic symbiotic association. Fungal roots were discovered by the German botanist A.B. Frank in 1855 in the forest trees such as pine. So basically 95% of the world's plant species, they form mycorrhizal relationship with fungi, and in the majority of cases the plant will fail to survive without such mycorrhizal relationships. So there are certain exceptions to this. For example, Brassicaceae, Cyperaceae, and Junicaceae. These plant species, they do not have mycorrhizal association. So 95% of them form the mycorrhizal associations. The plants, what happens in this such kind of associations, symbiotic association, is that the plant supply the fungus with the organic molecules such as sugars, which are formed through photosynthesis, and in turn the fungus will supply the plant with the water and the mineral nutrients which it takes up from the soil. So, as far as the classification is concerned, the mycorrhiza, on the basis of morphology and anatomy, they can be, classified into three categories. That is endomycorrhizae, ectomycorrhizae, and ectendomycorrhizae. So, let us first have a look at the endomycorrhizae. In this type of association, the fungal hyphae are present on the root surface as individual threads, and these hyphae will penetrate directly into the root hairs. Through there, they get the entry to the epidermal cells and also into the cortical cells. So basically, in the endomycorrhiza, there is an insertion of the fungal hyphae inside the root cells. They are also known as AM, that is arbuscular mycorrhizae. And in this arbuscular mycorrhizae, these are the most common and widespread of all the mycorrhizae that are formed in almost 85 to 90% of the world's plant species. This association is not a destructive parasitic association, but in endomycorrhiza, they are considered as a normal part of a normal root development in certain plants. So AM fungi, what it does is it penetrates the cell wall of the root cells. So basically endo words means it's inside the cells. Endo means inside. So mycorrhizal association inside the cells of the roots is endomycorrhiza. So the hyphae, the fungal hyphae, are entering the inside the root cells, the cortical cells, and they grow between the cell walls and cell membranes, forming the arbuscules. So the WAM fungi produces the vesicles for lipid storage. These are the specific types of endomycorrhiza which produces the vesicles which are specialized for lipid storage. This is an example of Endomycorrhiza where you can see the chlamydospore of fungal species, which gains the entry through the root hair, and from inside this they form these arbuscules inside the root hair cells. Examples of the fungi forming the Endomycorrhiza includes the endogon, glomus, sclerocytis, Achelospora, Gigaspora, and a few more. So, Endomycorrhiza can further be classified into five types. The first is VAM fungi or the vascular arbuscular mycorrhizae. Second is orchidoid mycorrhizae. Then you have monotropoid mycorrhizae, ericoid mycorrhizae, and arbutoid mycorrhizae. So vesicular arbuscule mycorrhizae is VAM mycorrhiza, which is formed with the plants that belongs to the Ascomycetes, Basidiomycetes, and Zygomycetes fungi. And all the VAM fungi are obligate biotrophic. That is, they will not be able to survive and they without the plant association, and they're completely dependent on the plants for their survival. So here in this figure, you can see the VAM mycorrhizae structure where through the root hair, the chlamydospore hyphae, they get the entry and they form the arbuscules inside the cells. You can see the young arbuscules which are smaller in size and the older arbuscules which have developed into the whole area inside the cells. So basically they penetrate the cell wall and the cell membrane, and they form the vesicles which are specifically their specificity is for the lipid storage, and also they form the arbuscules inside the root cortical cells. The second type is the orchidoid mycorrhiza, and the fungi that form such mycorrhiza belongs to the Basidiomycotina, and they colonize only the member of the family Orchidaceae, and this association is probably pseudomycorrhizal, but they play an important role in the establishment of orchid seedlings. So basically they are considered to be pseudomycorrhizal, but they are also important in the establishment of the orchid seedling. At some point of time, you can say that all the orchid plants will form the mycorrhiza. Okay, so the basically the family of Basidiomycota, this fungus will somehow at some point of time will form the mycorrhiza with the orchid plant, and they play an important role in the formation of the orchid seedling. Orchids germinate only after infection by mycorrhiza. Example of this is Rhizoctonia species. Within the cells, the hyphae forms the coils. These coils are known as phyllotones, and the function of the phyllotones is to increase the surface area, interfacial surface area between the orchid and the fungus, so that the nutrient exchange can take place with a higher rate. The second type of endomycorrhizae is ericoid mycorrhizae, and the fungal members which are involved in the formation of such mycorrhizae include the Basidiomycetes and Ascomycotina. Okay, so this type of mycorrhizae occurs in the family Ericaceae, where the rootlets are covered with the loose mesh of dark brown septate hyphae. From this, brown mesh, the branches or the hyphae penetrate inside the cortical cells, and the coiled hyphae fills all the cells and most of the cells in the cortical region of the roots. Up to 80% of the root volume can be fungal tissue, and it is through these coils that the nutrient exchange is thought to occur. So basically what happens in this kind of mycorrhizae, it occurs in the family Ericaceae, and the fungal species involved is Basidiomycetes, and there is a loose brown colored mesh of hyphae that are formed outside the rootlets, and from there the branches of the hyphae, fungal hyphae, penetrate or get inside the cortical cells, where the hyphae, forms a coiled structure, and they fill up the cells, where they forms the, coiled structure which is involved in the nutrient exchange. The third type of mycorrhiza is monotropoid mycorrhiza. This fungi belongs to the Basidiomycotina, which colonizes achlorophyllous members of angiosperms, which belongs to the family of Monotropaceae. So these plants entirely depend upon the fungus for carbon and energy, and there is a sheath, inter and intracellular hyphae, and the peg-like haustoria are present. The next type is Arbutoid mycorrhizae. Arbutoid mycorrhizal associations, these are the actually variations or the variants of Ectomycorrhiza, which are found in certain plants, and these are characterized by the hyphae coils in the epidermal cells. So the mycosymbionts are Basidiomycetes. The major difference between the Arbutoid mycorrhiza and the Ectomycorrhiza is that in the Arbutoid mycorrhiza, the hyphae, actually penetrate the outer cortical cells and fill them with the coils. Next, we are going to study about the Ectomycorrhiza. The Ectomycorrhiza or ECM, in this the fungi do not penetrate inside the cells, whereas it forms the mantle around the roots. So there is a formation of sheath-like structure which is formed outside the root cells, and the hyphae do not enter and do not form the coils inside the root cells or the cortical cells. There is no hyphal penetration inside the cell. So there's a difference between endomycorrhiza and ectomycorrhiza. That difference is that in endomycorrhiza, the hypha is penetrating inside the cell wall and they're forming the coils inside the root cells. Whereas in ectomycorrhiza, the hypha is not entering inside the cell and they're not forming the coils inside the cell. Whereas there is a form of mantle around the roots. The fungal hypha is generally separate, and for the exchange of the nutrients between the plant and the fungal hypha or the fungus, there is a formation of heart egg's nest which is present between the cells. Ectomycorrhiza or EM are typically formed between the roots of around 10% of the plant families, which belongs to the woody plants, including birch, dipterocarp, eucalyptus, oak, pine, deodar, and rose families, orchids. And the fungi that forms the ectomycorrhiza belongs to the Basidiomycota, Ascomycota, and Zygomycota. The ectomycorrhizal fungi, what they do is they form a sheath or a covering, or you can say a mantle around the root, and the hyphae emanate through the soil, increasing the surface area. The fungus grows within the root cell wall, but they never penetrate the cell interior. The fungus will never enter the cells. It grows between the cells of the cortex to form the heart net. In the cortex region of the roots, plant roots, they form the heart nest by growing in between the cells. The heart nest net, which is present outside the endodermis and meristematic zones, act as the site for nutrient exchange. Colonization of root tips induces marked changes in the host root morphology. The fungus that forms the Ectomycorrhiza includes the Amanita muscaria, Entomoloma, and Boletus variegatus, Bacillus involutus. So these are certain examples of the fungal species which are involved in the formation of Ectomycorrhiza. With the help of the diagram, you can understand that this is the simple diagram which shows the ectomycorrhiza. You have the vascular bundle, you have the cortical region, epidermis region, and here you can see that like the endomycorrhiza were forming the coils inside the cells. In this case, you can see this sheath which is present outside the root hair. Inside the cortical, in the cortical region, you can see this Hartix net which is present in between the cells, whereas there is nothing inside the plant cells. So the fungal species will never enter inside the cell. But if you look at the other diagram which shows the endomycorrhiza, I'll just show you again. In the endomycorrhiza, you can clearly see that the fungal hyphae are entering inside the plant cells. This is entering inside the plant cells, but in ectomycorrhiza, they are not entering inside the cells. They are forming the Hartix sheet in between the plant cells. So the advantages of ectomycorrhiza are extensive multi-branching hyphae, increases the water holding capacity of the cell. So because there is an increase in the surface area, increase in the branching of the hyphae, the water holding capacity of the plants increases sharply. Secondly, you have that increase the tolerance to drought, high soil temperature, organic and inorganic soil toxins, extremes of soil acidity to sulfur and aluminum. So basically the presence of mycorrhiza, or specifically you can say ectomycorrhiza, provides the tolerance to the plant from drought, temperatures, toxins and acidity. It also deters the infection of feeder roots by some root pathogens and enhances the uptake of nutrients, that is phosphorus, copper, zinc, through the Hartix net. They have an advantage of disease control through barrier effect and competitive exclusion, and they also play a key role in afforestation. Then you have the third category, which is known as Ectendomycorrhizae. Ectendomycorrhizae shares the features of Endomycorrhizae and Ectomycorrhizae. The fungi belongs to Basidiomycotina, which covers both gymnosperm and angiosperm plants. Ectendomycorrhiza show many of the characteristics of Endomycorrhizae, but they also have extensive intercellular penetration. So like Endomycorrhiza, they forms the Hartix net, and also like Endomycorrhiza, they forms the coils inside the cell. So the formation of Ectendomycorrhiza begins with the formation of Hartix net, which grows behind the apical meristem of the growing root. The Hartix net penetrates between the epidermal and the outer cortical cells and later extends to the inner cortex. The fungus which forms the Ectendomycorrhiza are Ascomycetes, and the host are Eucalyptus, Salix, Alnus, etc. So that is all about this topic.